Okay. Hello everyone. I'm Jongmo and the PhD student of the SEAP. I'm studying about the Russian though.
Thank you.
Hello everyone, and I'm Jongmo and the PhD student of the SEAP, and I'm studying about the Phoxinus species, especially in the Pyrenean lakes. And then I will start the presentations about the topic of the European minnow, the species and genetic variabilities in the national park. Okay, a long time ago, the people thought there is only one minnow species, that is the Phoxinus. But the way with the morphological data, people started to distinguish them, and in recent studies, they will reveal the distribution of the Phoxinus species using the molecular data. So in these studies can make us to understand of the introduction history of the alien species and make us to easy to control them. High mountain lakes are usually natural fisheries. They usually have their own native ecosystem, especially with the rich aquatic invertebrate and then some amphibians and reptiles. But after there have been numerous trout introductions to such ecosystems in many areas by fishermen. At the same time, the European minnows are also introduced as a bait for fishing the trout. So this species became invasive and drastically modify native ecosystems from benthos to native species such as brown trout. So they make so it makes negative effects on the local ecosystem. The recent study from the Danish in 2020, they discriminated the whole Phoxinus species from France. And they detected 6 species, and especially these 2 are new from the science. The interesting for me is that this species, the Phoxinus dragarum, because this one have been described after I studied about the Phoxinus. So the genetic analysis are revealing new species, and the continued research is needed. First of all these are primary part of my PhD study, so I apologize in advance about it because the result are quite in the first step of my PhD study. So the main objective is to figure out the genetic structure and the phylogeographic distribution of several populations of Phoxinus species from high mountain lakes of the national park of Aigüestortes y Estany de Sant Maurici, together with the other lakes from the Pyrenean lakes and the Alps. The total 33 lakes or rivers from Spain, France, and Italy are used for this study, and Aigüestortes y Estany de Sant Maurici National Park and Alto Pyrenean Natural Park from Spain, and the Grand Paradiso National Park and Mont Abic Natural Park from the Alps is our main site. They are main site. So if we see the locations on our map in detail with some nice photos of the lakes in the mountains, from the national park we have the Bassano del Lui, Estany del Lui, and then Sic de Trebassanis. And from the natural park we have 3 estanys, the Baix and the Mig and Crozer and Naurte and then Robinets. And also we have some sites from the high mountain lakes from the Aragon and from the France. And some of the rivers from the Catalonias are included here. For the genetic analysis, the DNA extraction was conducted by the hot shot and then Qiagen kit, depending on the condition of the samples. They were amplified by the PCR, and then the output were checked by the agarose gel electrophoresis, and the sequence was aligned and edited by the Mega7. So we're in the result. So total 341, 638 base pair co-sequences were used for these phylogenetic trees. So there are 22 Phoxinus species in this tree, and then 14 species with the names are described. They have been described so far before, and then the 8 species with the SP still not described yet. We have the 4 species from here. And the first is the Phoxinus chasqui from the Italian Alps, and then Phoxinus septimaniae from the Italian Alps, and then some rivers from the Catalonia. And most of the samples belongs to the Phoxinus dragarum, the new species from the last paper. Is from the whole lake from the Catalonia, and then some river from the Catalonia and too. And from the Aragon, we have a Phoxinus bigari. In case of the Phoxinus sumariae, it have been found from the Italian Alps in the other study, but we cannot find it from here, this study. So the next step is where they are from. They are native or they introduced. So if there are 4 species, to find out if the 4 species are native from the surrounding area, we make this figure to specify the native area of each species. So Phoxinus vulgaris originate native from the Adour basins from the French, and then Upper Ebro, and then some Cantabrian region from the Spain. And Phoxinus dragarum is native from the Garonne drainage from the France. And the Phoxinus septimaniae is from the Rhone drainage, and then some Mediterranean Sea coast side from the France. And the last, the chasqui, is from the most of the Danube drainages. So we can assume that if we can find these species the outside of this area, then that will be the result from the introduction. This is the result from the haplotype networks. And then we will start with the Phoxinus dragarum. So what is the haplotype networks? So haplotypes show genetically different individuals as suckers. The size of the sucker is proportional to the numbers of individuals found from each haplotype. So if there is some distance, more distance, it means they have more mutational steps. If you see the tables, you can see the number of the haplotypes from the Pyrenean lakes. So they have the 9 haplotypes, and it means at least they have been 9 different introductions. And also you can see the haplotype diversities from the table, and haplotype diversity is the probabilities that 2 randomly sampled areas are different. So as you see, the Garonne drainages, which is the native area of this species, has the most highest values compared with the other region. The next one is about the Phoxinus vulgari. So there is some interesting point. They are roughly separated by the 2 patterns. One is a little bit red patterns from the native area of the Adour basins in the France, and the other part is the others. The most interesting point is in here, this haplotype. You can see some individuals from the Ebro, and the other individuals are organized by the individuals from this Douro drainage from the France. So we can assume that this Phoxinus vulgari from in the Douro drainages are maybe introduced from these Ebro basins, and because they are in our native area, and they are also geologically very close. And when we see the haplotype diversities, it also see the same patterns with the others. So other drainages and the above drainages are native, so they have the most higher values, and the others are lower than that. The next species is the Phoxinus septimaniae, and this has the most complex construction of the haplotype network. And as you see, they are native from the Rhone and the Mediterranean coast of France. So when you see this haplotype network, you can see the individuals from some Catalan river basins. And the others are from the Mediterranean coast of the France with these red patterns. So probably that this Phoxinus septimaniae in this basin with the Catalan rivers are introduced from the Mediterranean coast of the France. So in the same theoretically, the haplotype diversity of the septimaniae from the native area is the most high, has a high values compared with the others. And the last is about the Phoxinus chasqui. So in this part is from the native area with the Danube drainages, and also there is a little bit part of the native area, and except that all the introduced part were gathered in here. So they don't have the many genetical differences compared with their native area. It also can show through the haplotype diversity from the table. So native area from the dam drainage has a almost like a double times higher than compared with the alpine lakes. And this is a summary of the 4 species. This is a summary of the native area and then current dispersion of the 4 species of the Phoxinus. So the chasqui is native from here, but now we can find that from here. And the Bulgari was a native from here, but now we can detect them from this area. Like Depth septimaniae is like this, and then Phoxinus ragarum is like this. So when you're considering the geographical distribution and result from the haplotype network, then we can assume that the minnows of the high mountain lakes are introduced from the other region. And this is a result from the microsatellite. This preliminary result using the microsatellite are only using separating them with the PCA, the 4 groups of the Phoxinus species. And the entry point was they separating even the same dragarum into the 2 groups. So this is the result from the microsatellite analysis. So each individual is represented by a vertical line, so which is colored according to the assigned groups estimated by the K value. So colors represent the probability of the ancestry to each cluster. So we can find some So we can figure out how many introductions are happened in this species, and also we can check I'm sorry. Also, we can know the percentage of the species gene flow introduction from the species. At first, we conducted a first analysis, so they separating the whole individual into 2 groups, with the orange with the A and the blue with B. So we conducted a second analysis with the separating each group. So from the group, class A is divided into 3 groups. So Phoxinus septimaniae, Phoxinus bigari, and Phoxinus dragorum. But even the same Phoxinus septimaniae, there is a clear division with the septimaniae from the Alps and then the septimaniae from the rivers. And the class B is divided into 5 groups, even if the horde of Phoxinus dragorum species. So we can see it's divided by their own site. And if there is a variety of the colors in a one vertical bar, means there is the many introduction cases for this site. So we can see in case of Sigde travasani, there is a lot of introductions from their population for this population. But in the high mountain lakes, it's hard to introduce naturally from lake to the another lake. So maybe it's a result from the human activities. This is a result from the DAPC. So they separating the whole group into the Phoxinus dragorum and then Phoxinus septimaniae and bigari, and then Phoxinus dragorum from the Delui. This result is about the recent gene flow from the gene class 2. So this 85% of the individuals they are assignment from the populations, but the 40.8% of the individuals are exclusion of their own population. So it means they have a lot of the introductions from the other side. This program also can calculate the migrations. So 23 individuals was figured out, was detected for the migration, and then 17 is from lake to lake, and then 1 is from the river to river, and 4 is the lake to river, and then 1 is the river to lake. So the translocation of individual is a potentially present phenomenon, and then in the high mountain lakes and rivers. So further investigation is required for the full confirmations. So we detected 4 species from the high mountain lakes and surrounding areas, and most of the individuals were Phoxinus dragorum. And by the haplotype diversities, the minnows of the high mountain lakes were introduced from the other regions. The translocation of the individuals is a potentially present phenomenon in high mountain lakes and rivers, so additional study is required. Progress of multiple introductions have taken place in the Pyrenees. In addition, we have found several genetic lineages within some lakes indicating multiple introductions or events. Thanks for reading, and sorry about my terrible English.